all of you. Welcome once again to the NEET Bytes session presented to you by Meritnation. And I can see many people out there. Hi, Shravya. I guess I'm taking your name correctly. Hello, Aditya. Hi, Aman. Hello, Shakti. So, how are you doing, guys? What's about your preparation? And what are the topics you've been covering? I hope you are following the schedule and you are trying your best to cover up as much as you can. So, as you can see, we are back again with respiration once again. Okay. In the previous session, I told you that we will be covering respiration in 2 sessions, right? But it's a big topic and conceptual topic, right? So, in the previous session, we have talked about aerobic respiration. In today's class, we'll be talking about anaerobic respiration. Remember, we have divided the respiration, we divided it into 2 parts. Okay. We divided. Yeah. Or is it divided by nature? So some of them are doing aerobic respiration, some organisms, and the rest others are performing anaerobic. Okay. So in today's class, we'll be focusing on this guy. Okay. What is this anaerobic respiration? As well as we will find some questions about ETC as well. Okay. So welcome Meghna, welcome Sunny, and welcome Gurbaksh, welcome Gurpreet, Sim name. I welcome all of you. Okay, so let's first of all see what is anaerobic respiration. Okay, how is this guy different from the aerobic respiration? A little bit of the topic we need to see here. Okay, so what is the difference? As the name indicates, anaerobic respiration will not involve oxygen. Where? Let's find that out. Okay, so be it aerobic respiration, be it anaerobic respiration, both of them, they start with the hero glucose. Right. Okay. And the glucose will undergo some process. It will form pyruvate. Okay. You know this, right? Can anyone tell me what am I doing here? Can you tell me what is the process I'm talking about? Yes? Can you tell me which process I'm talking about? What is this process known as? You know this. This is glycolysis. Okay. Glycolysis is the killing of glucose. Right. It happens in both the types of respiration. Okay. But in case of anaerobic respiration, this is the only process that yields energy. Okay. Mind this very carefully. This is the only process that gives out 2 ATPs. Okay. This is the only thing. Rest, you should know that glucose cannot be converted into pyruvate until and unless you have this person present over there. There must be the presence of NAD so that it can accept the electron and it can be converted into NADH. Okay. Now think about an anaerobic person. Okay, think about an anaerobic person who is converting glucose into the pyruvate again and again. But this will not happen until and unless you provide NAD back again. If all the NAD are consumed, glucose will not be able to be converted into pyruvate. So that's a bad idea. We need to regenerate NAD here. It's an electron acceptor for the glucose. Okay, so what happens depending on how we regenerate the NAD, okay, how we are going to regenerate NAD from NADH, depending on that, there are 2 processes that can happen from here. Okay, are you making sense what I'm saying? So your pyruvate can be directly converted into lactate. Right. That's called lactic acid fermentation. Okay, and this step is catalyzed by an enzyme called lactate dehydrogenase. Okay, lactate dehydrogenase. And in this process, what do you get back? You will get back your NAD. Okay, your NADH will lose the electron and you will get back NAD here. And this NAD will then be used back again here. It will be used back again here. The second process that can happen alternatively in some organisms, it happens, and you will be telling me in which organisms it happens, that pyruvate will be converted into acetaldehyde. Okay, it will be converted into acetaldehyde. And from acetaldehyde, we are going to get ethanol, the alcohol. Okay, right? Okay. Now, this particular process is a decarboxylation. So, of course, we are going to have the release of CO2. Your CO2 will be released here. And this step is where you are going to get back your NAD. This guy comes back here and you will be using it back again in the glycolysis process. Okay. So, can you tell me the name of organisms where lactate fermentation happens and name of organisms where ethanolic fermentation happen? Come on, guys. May I know that? So I'm getting an answer. Yeast from Aditya. So yeast is doing what? Lactate or ethanolic fermentation? Lactate or ethanolic fermentation? Right, right. Mussels and yeast. What are they doing? Okay, so exactly. Lactate fermentation, that's happening in your mussels. And this ethanolic fermentation is occurring in the yeast. And that's why you have all the bakery going on, isn't it? Exactly, yeast for ethanol. Okay, very fine. Perfect, Aditya. That's right. Exactly, Shravya. That's good. That's right. So this is an overview of your anaerobic respiration. So what have we learned from here? We have learned that it is only glycolysis which is making ATP. Okay, it is only glycolysis that makes ATP. Otherwise, the fermentation processes are just occurring so that you can have the NAD back again. They're not making any ATP in that process. Okay, right? Okay, so having studied that much, let's move into the question. Let's try to solve some question based on the topic we have seen. Right, guys? Are you ready for that? Shall we move on to the questions? And remember, this is going to be a quiz session. So we will be having 1 minute destined to solve the question. Within that minute, you will be providing all the right answers. That's what's going to happen here. So let's move quickly to the very first question. Okay, your time has already started. This is one easy question. And it is asking you that during anaerobic digestion of organic waste, okay, that sometimes we use for making biogas, which of the following material is not degraded? It is left undegraded. Okay, can you think of that? Is it cellulose? Is it lipids, lignin, or hemicellulose? Now, this is an easy question. Why am I saying that? You have to apply your brain and think that which one is a tougher particle, sorry, which one is a tougher material, which one is easy to break down. Okay, something that your bacteria cannot break. Okay, here you have to think of the substance that your bacteria cannot break. Your time has already started, people. Come on, birth out the right answer. The time is about to get over. Quickly, everyone. And this is one factual question. Doesn't include much of your conceptual knowledge in here. Okay, so your time is up. Your time is up. And yes, all the people who have given C as the answer, perfect. Yes. So lignin is not broken down during the anaerobic respiration. Okay, because you must understand that if something of your anatomy you remember from the plant section, lignin is responsible for providing the sturdiness to the plants. Okay, it's not broken down. The process of breaking lignin is something called as lignin pyrolysis, and that involves your fungus rather than your bacteria. Okay, otherwise your bacteria are like pro in killing the cellulose, the lipids, and hemicellulose. They can handle it, but not the lignin. Okay, so this was an easy question. It was one easy question, right? Let's move on to the next question, everyone. Heading towards the next question. Time has started. The question is asking you about oxidative phosphorylation. Perfectly. Okay. It says, what is it? Simply, right? So is it the formation of ATP by transfer of phosphate group from a substrate to ADP? Do you think that? Oxidation of phosphate group in ATP. Is it what you call as oxidative phosphorylation? Addition of phosphate group to ATP, or it can be formation of ATP by the energy released from electrons that are removed during substrate oxidation? Think carefully. It's a one roller coaster question. Okay, think and answer. The time is about to get over. I'll provide 10 seconds extra for this question. Everyone, buff out the right answers. All of you, quickly. Everyone must be writing the answers. Come on. You have like 10 seconds more to go. Think and we'll discuss this. Okay, think and we'll discuss this one. Right, right. Okay, your time is up. Your time is up. Let's read this question. See, the question is asking you about the oxidative phosphorylation. Okay, had it been about the substrate level phosphorylation, okay, had it asked you about the substrate level phosphorylation, you would have gone for this guy. Okay, you would have gone for option A, right? Are you making sense what I'm saying? But since we are talking about oxidative in here, what happens is that all the electrons that are released during the oxidation of substrates, they are caught, okay? They are caught by this guy, okay, NAD, and it gets converted into NADH. And then we are going to use this person in the electron transport chain, and therein we are going to make the ATP. Okay, so this option makes more sense than the first question. Okay, these two options are not holding any conceptuality in them. Okay, it's only about the first and the last question option that is giving a little bit of confusion here. So remember, guys, the first option is giving you the definition of substrate level phosphorylation. Simply, we are taking out the phosphate group and providing it to the ADP from a substrate. That's something happening during the substrate level phosphorylation. Okay, so be careful in these type of questions. This was one easy question. I guess you've understood. One easy and bit conceptual question. Okay, is that understandable? I hope it is. Okay, so moving on to the next question. Right, everyone? Shall we switch on to the next question? Okay, shall we move on to the next question? Perfect. Okay, time has started. The question is asking you that in which one of the following processes CO2 is not released? Okay, so if you little bit remember something I just told you in the beginning of the class, there is a step where we don't release carbon dioxide in the anaerobic fermentation. Okay, it's one type of fermentation. It's occurring by two processes. In one type, we are not releasing carbon dioxide. Okay, think about it and answer. And it is just because of the enzyme. Okay, it is just because of enzyme that no oxygen is released in this process. The correct answer happens to be because of the nature of the enzyme involved here. Okay, so I can see people trying to answer. You have few more seconds to go. Let's see what comes up, what we have in store. Okay, your time is up, guys. That's good. And yes, you all are correct. Lactate fermentation does not give out carbon dioxide. Why? Because of this enzyme. Okay, this enzyme does not do the decarboxylation. Okay, lactate dehydrogenase, it doesn't perform the process of decarboxylation. Carbon dioxide is not released. That happens in the case of alcoholic fermentation. Okay, if you talk about alcoholic fermentation, then yes, right? What happens in here? When you are converting pyruvate to the acetaldehyde, when you do that, then remember it's a decarboxylation reaction. Okay, it happens due to pyruvate decarboxylase enzyme, and there we have the release of CO2 happily. Okay, but not in this case. No CO2 released here. Okay, fine. So keep this in mind. Easy question. Let's move on to the next question, everyone. Okay, let's switch on to the next question. Are you ready, guys? All of you? I don't see much enthu today. What happened? Is it because of anaerobic respiration that you are not having much ATP in you? It's not like that, okay? I mean, you are studying about anaerobic respiration, but you don't really have to feel it by your heart. You guys are doing aerobic respiration, okay? So come on, you need to show that you are making lots of ATP. Everybody, be active. Use your keyboard. Why do you have it? Okay, next question. The time has started. The question is asking you about a beautiful concept of chemiosmotic coupling hypothesis. Wow, wow, wow. What is that? Okay. It is telling you that this particular hypothesis which tells us about the oxidative phosphorylation, proposes that adenosine triphosphate is formed because. So, which factor is taken into consideration when you talk about chemiosmotic coupling hypothesis? One easy question. Is it because of high energy bonds that are formed in mitochondrial proteins? Something like that. ADP is pumped out of the matrix into the intermembrane space. Proton gradient is forming across the inner membrane. There's a change in the permeability of inner mitochondrial membrane towards ADP. Which option seems to be legit? Which option seems to give you the right answer? Your time is about to get over and it's moving towards the finish and it's finished. Okay, so I'll draw something here. Okay, I'm going to draw a section of your mitochondria. This is the outer membrane, this is the inner membrane. And you guys know that your inner membrane of mitochondria is curly person. It has curls in it. And why? Because there are things present here. All of your electron transport chain is performing in here. What exactly happened is that when the ETC is moving on, when the ETC is in the process, the different complexes which are involved in ETC, they are transporting proton ions from the matrix of your mitochondria to this space. Okay, they are moving it out. I'm just giving you a pictorial representation. Do not take it, this is not the exact thing. Okay, I'm not making it via the complexes. I'm just trying to give you a pictorial representation. So when they do it, all the proton ions, they get accumulated here. And you know that nobody likes to stay in one place, isn't it? Nobody likes to stay in one place. They're like, wow, we are having high concentration here. The concentration is quite high here. The concentration is low here. It's a party time. We will have to travel. Okay, we'll have to move from this place to this place. Okay, but when they all are ready to move, there is this person. This person is called ATPase. It says, I am going to pass you down there. You all will not move on your own. I will guide you to the matrix. So all of them make a line and they stand. And this guy is a very cunning person. You know why? When these protons are going to move through the ATPase, this guy will use their force and it will make ATP. You see that? The poor protons are getting used. Wow, isn't it? So it says, wow, I'm going to let you down and I'm going to make ATP. Yeah, the process is done. Okay, so this is just like you have a pressure cooker. Okay, and lots of pressure is being built inside because of the heat. And then finally, CT will be there. Okay, it will whistle out. Exactly in the same manner, in this space of mitochondria, lots of protons are built up. Okay, and when they become lots in their concentration, they will move down through the ATPase and they will result in the formation of ATP. This is your chemiosmotic coupling hypothesis. It is because of the proton gradient, which is more in the perimitochondrial space and less in the matrix. Why? Because of ETC happening. Okay, because of ETC. So I guess this makes some sense to you. Okay, let's move on to the next question quickly. Right? Fine. So here comes your next question. Okay, your time has started. Question asks that during which stage in the complete oxidation of glucose are the greatest number, okay, the greatest number of ATP molecules formed from ATP. In the previous session, I gave you the summary of respiration. Aerobic respiration, I gave you the summary in previous session. If you guys were not there, please make sure you see that. I told you that how many ATPs you are making in each step. Okay, some of you who were present in the previous session, you guys remember that. So now it's the time that you put it into the use. You will be telling me in which process exactly do we form the maximum number of ATP in here. Okay, the time is about to get over, guys. Answer, everyone. Buff out the right answers. Time is about to be over. Okay, let's see who all managed to give right answer in this question. Right, so your time is up. And correct answer is electron transport chain. Yes. Remember, all the NADH that you have made in the glycolysis, in the Krebs cycle, and the AVDH2 that you have made, in the Krebs cycle, they will be used, they will be robbed of their electrons. Okay, they will be robbed of their electrons, and those electrons will move across the electron transport chain, and they will be finally accepted by whom? Who is the final acceptor in aerobic respiration? Finally accepted by this person. Okay, that is why it is aerobic respiration, for God's sake. Okay, the person will finally accept that. And while your electrons are moving down the ETC, they are causing the movement of protons. And those protons will then be robbed again. Okay, they will be robbed again by the ATPase to make the ATP. Okay, so there's lots of robbery happening in your cell. That's lots of robbery occurring in your mitochondria. Okay, sometimes they are getting robbed, and sometimes the protons are getting robbed. Is that clear? Does it make sense to you? I hope it does. Fine. Now, moving on to the next question. Right, people? Shall we move on to the next question? Perfect. Time starts. The question is asking you that in alcoholic fermentation, what happens? That oxygen is the electron acceptor. Do you consider this legit statement? Triose phosphate is the electron donor, while the acetaldehyde is the electron acceptor. Triose phosphate is the electron donor, while pyruvic acid is the electron acceptor. There is no electron acceptor at all. What happens during alcoholic fermentation? Now you know that electrons have to be accepted by somebody. Why? Why do you want electrons to be accepted? Because you want your NAD to come back. Okay, you want NAD return. Okay, this is your motive, right? And for that matter, you need to rob NADH of electrons. Somebody must be there to accept electrons from the NADH. So come on, put out the right answers. I'm giving you 10 more seconds, everyone. Think and answer, all of you. Think and answer. Okay, your time is about to get over, and it's over. Okay, yes, so I can see many people trying to put up the right answer. Let's see what it is. So exactly, give me a few seconds. Yes, exactly. B is the correct option. What is happening, right? In here, if you remember, I told you in the very beginning also that pyruvate is forming acetaldehyde. Okay, but that is a decarboxylation reaction. Okay, here we are removing carbon dioxide. Right. Now this acetaldehyde, this guy will accept the electron and it will turn into an ethanol. Right. And during this reaction, what happens? Yes, during this reaction, you are going to get back your NAD from the NADH. So this person here is the electron acceptor. And triose phosphate is not an alien. We are talking about pyruvate here. Okay, it is just the pyruvate which is written in its chemical form, triose phosphate. Okay, so this was just a trick to confuse you guys. Okay, and I can see lot many of you got confused as well. Okay, moving on to the next question. Shall we move on to the next question? Okay, and this is going to be your last question of the session. Okay, ATP is injected in cyanide poisoning because it is. So if cell suffers from poisoning caused due to cyanide, we have to inject the ATPs. Why exactly should we do that? So it makes sense, this question makes sense that cyanide is playing with ATP function. It is doing something that is not allowing ATP to perform its function. And that is why you need to insert ATP so that ATP, which are externally inserted, can do their activity. Okay, so is it because ATPs are responsible for cellular functions? Is it because they are necessary for sodium potassium pump? Is it because the sodium potassium pump operates at the cell membranes, or the ATP breaks down during cyanide poisoning? What happens? What is the right answer? So this is a very easy question. You know what is ATP doing for you? What is ATP doing for you? Yeah, it's making Maggie for the cell, right? It does that. Yes, of course. So if the cell has to be prevented from death, due to lack of Maggie, you have to make ATP insertion externally. Okay. So I can see people putting up the right answer. Okay. Let's see if you all have put in the right answer over there. So your time is up, everyone. Right. And yes, correct answer is option A. So if cyanide poisoning is happening, it interferes with the functionality of pumps, those which are required for the formation of ATP. And if there will not be any ATP, the cell will die. The functions will not be performed. Okay. That is why we need to inject the ATPs into the cell. Okay. This is one question regarding the importance of ATP. Okay. Now, with that, we have finished our session. Okay. I hope it has given you a sort of revision for your anaerobic respiration. In a short while, I'm going to be announcing the names of people who have given the right answer. Okay. So just a bit of wait, and you'll have the names of the people who have given the right answers. Yes, Meghna. Yes. In a short while, we are going to have the names. So just wait for a few seconds. There will be an announcement. Okay. So, people, what have you learned today? Yes. What have you learned? What is the important concept you got from here? Is that you need to revise. This is our default concept. Okay. Whenever we are finding it difficult to solve some concepts, solve some question, the very first thing that we learn is that we need to revise. And these are not very difficult things. Okay. Easily manageable. Right? Okay. So definitely, we are going to look in that. Fine. Thank you, Meghna. This is highly appreciative. Okay. Anything else that you guys need to talk about? Yes? Anything that we need to talk about? Okay. All about NAD. Yeah, exactly. So why is that so important, Nusrat? Okay, Nusrat, that's your name. Okay. NAD is an electron lover. Okay. Sometimes in the cell, we have to kick out the electrons. Sometimes in the cell, we need to have the electrons. Okay. Whenever you have to kick out the electron, you'll need this guy. Okay. You'll need this guy. And whenever you want to have the electrons, you will need this guy. Okay. So this person keeps on alternating between two forms. Sometimes it is rich, sometimes it is poor. It is rich when it is NADH. It's like carrying electron. Yo, I'm having electrons with me. When it doesn't have electron, it is NAD, and it's always looking out for electrons. I want electrons. I want electrons. Okay. That is why it's all about your NAD. Okay. So people, I think there is some technical error today that we have failed to get the names of people. I'm regretting about that. I'm sorry for that. I'm sure this will be solved by next class. Okay. So it's time for me to say goodbye to all of you. Okay. And you all know how many of you have given the right answers, how many of you were comfortable with solving the questions. Literally, Aditi, highly, highly sorry. Do we say that? Right. I'm very sorry for that. There were some technical issues, maybe because of which we haven't gotten the names. Fine. But not a problem. In the next session, that will be corrected. Right. And so I'll take your leave. We will meet again in the next session. Stay connected, keep revising, and we will meet again with more questions, more concepts, more quizzes. Okay. Till then, all of you be good, stay in good health, keep studying, keep revising. Bye-bye. Bye to all.